Hi, my name is Tricia Shoemaker, and my presentation is on CRISPR-Cas9 gene editing for sickle cell anemia. So we're going to briefly go over the background of sickle cell anemia, the current testing methods, current treatment methods, CRISPR-Cas9, the FDA-approved CRISPR-Cas9 treatment, and the clinical trials and future use of CRISPR-Cas9, and then my references. So sickle cell anemia and its associated disease is a group of disorders caused by a point mutation in the hemoglobin S allele. Most common is a homozygous point mutation in the codon coding for glutamic acid, where thymine is substituted for an adenine in the codon for glutamic acid, resulting in valine and rigid sickle cell shaped blood cells. The heterozygous state of sickle cell disease is sickle cell trait. Patients are mainly asymptomatic except in rare occasions. Other sickle cell disease variants can occur where glutamic acid is replaced with lysine or glutamine. Similarly, beta thalassemia, which is an imbalance in the alpha and beta globin synthesis, reduces or eliminates beta globin production. And so the patient can be seen with anemia. Cell mutation destroys the restriction endonuclease, thus cutting the nucleic acid in the beta chain. This makes identifying the patient's status straightforward, as we can utilize BSU 361 to cut it into fragments. The most common method for testing is real-time PCR. The genes for hemoglobin sit on both chromosomes 2 and 11. For sickle cell disease is hydroxyurea. Bone marrow transplant from a sibling is sometimes done, but is usually the last choice due to graft versus host disease. Two newer drugs on the market help increase fetal hemoglobin. Those are voxelotor, which increases hemoglobin's ability to bind oxygen, or crizanlizumab, an antibody to P-selectin, which decreases the cell adherence to the endothelium and increases blood flow. Gene therapy using Lyfgenia or Casgevy are two of the newest drugs on the market. They are a one-time infusion that is given to the patient and is usually administered for anyone 12 years or older. The patient is first placed on plerixafor to stimulate the CD34 positive hematopoietic stem and progenitor cells. The HSPCs are collected, genetically cryopreserved, and then tested. The patient undergoes myoablation, usually with bisulfan. And then the cells are thawed and infused into the patient. Two gene correcting techniques have been approved to date. One is lentivirus-based vector, Lyfgenia, and the other is a genome editing method using CRISPR-Cas, which is Casgevy. Both were FDA approved in December of 2023. Genetic editing involves altering the gene sequence directly. Modifications contain a DNA binding molecule and an active epigenetic modification molecule. DNA is cut using endonucleases to make a double-stranded break and allow the ends to repair. The new sequence uses a template delivered into the cell with the endonuclease to repair the sequence in the area of the cut. The three most common binding molecules are zinc finger proteins, transcription activator-like effectors, and deactivator deactivated Cas9. CRISPR, or clustered, regularly interspersed short palindromic repeats, and Cas are arrays of regions in bacterial genomes, genome DNA used to store genetic information from infecting viruses. The bacteria uses the genome to transcribe RNA sequence to cleave the homologous region of the viral genome upon reinfection. Electroporation, or mRNA, can be used to allow Cas9 into the cells. Virus-based methods can also deliver the donor template. Lastly, non-viral delivery via single-stranded oligodeoxynucleotides can be used. Genome editing strategies have been designed to either correct the mutations responsible for beta hemoglobinopathies and restore endogenous HBB expression, or induce high levels of endogenous gamma globulin expression. Three different methods using CRISPR-Cas9. A, which is CRISPR-Cas, can be used to cut out the gene error in the beta globin gene using donor DNA template. B, which is inducing expression of the HBG Macassar variant by substituting guanine for adenosine. C, inducing an indel of the erythroid-specific enhancer BCL11A, which is located on chromosome 2p16 to depress the gamma globin genes. This induces fetal hemoglobin by disrupting the GATA1 binding site in BCL11A. D, using CBE to disrupt GATA1 in the BCL11A, reactivating fetal hemoglobin. E, which is induce indels in hemoglobin 1 and 2 promoters to disrupt the fetal hemoglobin binding sites and reactivate it. Or F, induce mutations into the gamma globin genes. Casgevy's mechanism of action is the activation of fetal hemoglobin by inhibiting BCL11A, which was example C on the last slide. Casgevy has been on the market for a year. It is indicated for individuals 12 and older who have had recurrent vaso-occlusive events. It has also been approved for beta thalassemia patients by using the CD34 positive HSPCs edited with the CRISPR-Cas9 and a guide mRNA. Utilizing CRISPR-Cas9 does have some disadvantages. Most patients experience at least some of them during treatment, although most are of grade 3 or 4 severity. Off-target effects, which might modify the expression of non-targeted genes, is also possible, but has not been yet seen in Casgevy. Hypersensitivity reactions to the cryopreservative solution have happened. Delayed platelet engraftment can occur. There are also some medications commonly used in sickle cell disease patients which can interact with Casgevy. Finally, pregnant patients or those who have potential to become pregnant should not use Casgevy. There are several ongoing clinical trials for sickle cell disease using CRISPR-Cas9. You can see here that we have Casgevy listed. These other clinical trials are still underway. So the future of CRISPR-Cas gene editing is bright. I anticipate additional clinical trials using other gene editing techniques in the future, including base editing for other diseases caused by known indels for cancer therapy. It is important to remember that utilizing CRISPR-Cas9 does have some disadvantages. Most patients experience at least some of them during treatment, although most are of grade 3 or 4 severity. Off-target effects, which might modify the expression of non-targeted genes, is also possible. Most of these can be limited by using high-fidelity Cas9 nuclease. So here's a list of the references for my presentation. listening to my presentation. It was really interesting doing research on all of the new uses for CRISPR Cas9 gene editing. We used it when I was in college on E. coli, but it was blind, and so it's nice to see how it's used in a more effective way.